We're very pleased today to have with us Larissa Stanberry, Dr. Larissa Stanberry, from the Minneapolis Heart Institute Foundation. I met Larissa when I was a postdoc at the University of Washington. She was finishing her PhD with B. Julian Lisak and Tillman.
Tillman,
19. And afterwards, she went on to a position at the University of Bristol. But since then, she's come back to her biostatistical roots and is currently at the Minneapolis Heart Institute Foundation. She's the program director of data science and investigator-initiated research. So please join me in welcoming Larissa.
Well, thank you, Hannah. Thank you for inviting me here, and thank you for everyone for hosting. It's been a while since I visited Toronto last time, and I don't think I've been here this time of year. So usually it's the summer, what I'm doing. It's very nice to see the different seasons. Couple of things first. I may cough through the presentation. As I explained, I'm not contagious. I caught something back in January, and so once in a while it comes back. So bear with me. And I'll try not to cough into the microphone. And feel free to interrupt me at any time and ask questions, please. I have a bit of an accent, so if you don't understand, then also wave and ask me to slow down or repeat myself again. So all good things. Yeah, exactly, get started. So this work is a very fresh on the press. It's actually still being printed as I speak. It's something that was been on my mind for a while, and I just decided to take a deeper look at it. And so it's my first kind of showing it off to the world. Really looking forward to your suggestions and comments and ideas you may have. So a lot of this project was done with support of my team. Everybody is listed there, so they all from the Minneapolis Heart Institute Foundation. Everybody is a statistician except for Andrew, who's a computer scientist, and does our data management and support. All right. So a brief agenda for the talk. I'll tell you a little bit about the foundation where I'm coming from, because it's a pretty unusual organization. And I think when I was in grad school, I kind of thought that it's either industry or academia, and the world seemed very black and white. And then there is a very different niche of research organization. And so I'll just tell you a little bit what the MHF is, and just so you know a little bit more where I'm coming from. Then I'll speak to you about the motivation for this particular research problem. I'll give you a brief refresher on predictive modeling, and then talk about the study design and results. Right. So the Minneapolis Heart Institute Foundation, we're an independent nonprofit cardiovascular research organization. We were founded in 1982, the leading life-saving cardiovascular research and education to improve care for patients around the world. That's our very ambitious mission. We have a little under 100 people on staff. That's not counting PIs. So it's the research support staff, about 20 million operating expenses annually, and some amount in net assets. The foundation was founded a year after the cardiology practice Minneapolis Heart Institute came to be. The cardiology research in Minneapolis area is probably best kept secret. You guys know Dick Cheney. Ever heard of that guy? So his first cardiac intervention was done in Minneapolis. I found if you know what a diskette is, I found a little floppy drive in my first desk. That's a Dick Cheney. It wasn't a computer created, but we do a lot of biotech research is happening in Minneapolis, in Twin Cities areas. You know, the pacemakers were invented in Twin Cities, and so there is a very vibrant biotech community in Twin Cities. So the cardiology practice is one of the best in the country, and we participate in multiple clinical trials and carry some flagship research with various industry sponsors. So it's about 100 plus cardiologists and surgeons. And cardiologists come in all kinds of different flavors. So if you have any interest to learn more, I can tell you all the different flavors that I came across. 40 sites across Minnesota and Wisconsin, so various clinics and hospitals. And so we do clinical practice with research. and we do the education. We're not part of the line of health system, but we, as a foundation, we're not part, but we work very closely with MHI because that's where our PIs are coming from, who are in some kind of relation with Alina House. And that's kind of complicated, so I can tell you more than that, but that should be enough. So we have a pretty sizable study portfolio. So just on our investigator-initiated research, we have at any given time about 100 very active studies open. The study portfolio is split 50-50. So we have the research projects that come from our own PIs, and then we have industry sponsors who come to us and say, well, we're interested for this market studies for this particular device or clinical trial, can you help us enroll patients? So my group does not deal with the industry sponsored clinical trials. This is a completely separate part of the operations where our research coordinators happen how to identify the patients, enroll the patient, consent the patient, collect data, and send it to the industry sponsors. What we're working is with our study PIs who are interested in pursuing some kind of research topic. A lot of it is sort of how can I improve patient care? So it all evolves around that big concept. And it varies from understanding long-term implications for current therapy, trying to distinguish if there is any differences between, say, two equivalent therapies, seeing long-term outcome for existing well-established interventions, as well as new interventions that come into practice, right? Because a lot of research, like a lot of randomized clinical trials, they actually have a very narrow window for the follow-up, typically looking at, say, 1 year, maybe 2-year events, before getting FDA approval for the technology and moving it forward. And so nobody really knows when they introduce a new technology or new drug or new treatment to the market, how well it will perform in 5-10 years from now, right? And that's what our researchers are interested in. So there's a million questions around clinical practice, all that come from the patient bedside eventually. And so they come to us and we help to deliver, to make this project happen, so to speak. We also have a very vibrant internship program. So every summer we have about a dozen students who come and do research projects with us. It's, the program's been running for the past 20 years. We also do various community outreach, educational events, student-patient education. In total, we have up to 2,500 publications across all the PIs. And then they go to some of the big journals in the field like JACC and JAMA and so on. Now, for this particular research project, it really came from my experience as a statistical editor for the 2 journals of American College of Cardiology. So ACC has a whole line of journals. They started with a 1 journal, JACC, and then over time, as the demand grew, they kind of expanded and made more specialized journals. So I'm the statistical editor for JACC Heart Failure and JACC Advances. The time they're using JACC EP, I think, when they get overwhelmed. But as I, read through those papers in the last couple of years, I noticed a certain trend. I noticed that people trying to bring more and more predictive modeling into what they're trying to publish. And it's not always well done, and it's not always well justified. And at some point, it feels like it's really kind of like one of those things that people put at the end of the paper for no particular good reason. And I just started to wondering if it's just me kind of imagining things or if this is something that's really happening in the field. It seems that the quality is lacking while the quantity persists. And very often there is lack of understanding what is inferential modeling design, what is predictive modeling design. And I don't see a whole lot of statisticians involved in a lot of those papers. So anyway, I decided to kind of dig in a little bit and understand if that is truly a phenomenon. And so the project goal was pretty simple, right? So review the published clinical studies, look at those that are presented as prediction modeling, evaluate the quality conduct of the study. And as a case study, I decided to look at the predictive modeling in heart failure. So before I joined the foundation, I thought, the one thing to die is a heart attack, right? But after there's the foundation, well, there's like a whole line of different cardiovascular diseases and things that can go wrong with your heart. So kind of a knowledge you should know in times, but heart failure is one of those. And heart failure is essentially something happens with the heart. Usually it has something to do with coronaries. And so coronary arteries are those arteries right on the top of the heart that you see. And the mission essentially is to feed the heart itself. And so those arteries tend to narrow over time. They can get calcified. It can be due to age. It could be due to some, family history. It could be socioeconomic factors, race. But as the arteries, the coronary arteries sort of change, that affect how the heart functions, right? So it doesn't pump as efficiently. It starts enlarging, and then it starts sort of lead to all kind of different side effects, right? So you get a lot of swelling, shortness of breath, there's fluid accumulates in lungs. Over time, it can lead to multi-organ failure. And the way to sort of help with this disease is either heart transplant or there's a left ventricular assist device that gets implanted and gets heart pumping. And then, of course, it is pretty dangerous condition. The treatment also has a variety of adverse events associated with it, right? So it seems like a big disease. Now the question is, why did it pick this particular disease? Because other diseases, as I said. Now the reason for that is heart failure it has a huge prevalence. So the stats here for the US, and my guess maybe numbers are somewhat similar in Canada, but it's almost near 7 million adults with heart failure in the US now, and the number expected to increase. 1 in 4 expected to be diagnosed with heart failure in a lifetime. There's a lot of morbidities and mortality associated with a heart failure. There's huge medical costs, right? That includes healthcare, prescription, missed work days, and then the incidence and prevalence of the heart failure and adverse events associated with heart failure kind of varies depending on the age, race, location, so those factors. And because disease is so prevalent and so well known and it's been around for so long, there is a lot of clinical research that was done on health in heart failure. So that, for my particular project, that means I have a lot of data points, right? Or I hope to find a lot of data points. Now, there's a number of prediction models that have been published. The adoption and practice is quite slow. So there's more publications than the scores that are actually being implemented. There's a number of publications that looked at how these models were developed. Some were found to be well validated and maybe have some clinical value. The latest Titana, they looked at papers published, I want to say, I'll tell you exactly, but about like five years of paper being published. So they found 40 risk models, and they found that the low risk of bias is only 20% of the models, right? So they essentially the saying that majority of the models are kind of biased. So now Ditana uses particular probus tool, and I'll tell you a little bit more about this probus tool and why we chose to do what we did. But before we do that, I'll tell you a little bit like a quick reminder about the prediction models. And so prediction models can be roughly classified into two groups. So it can be a diagnostic prediction model or prognostic prediction model. So for the diagnostic prediction model, the heart failure, we typically think about maybe diagnosing disease based on biomarker levels, or distinguishing different subtypes of heart failure based on patient characteristics or the biomarkers. And the prognostic models, so those are the models that can be developed in patients with heart failure, as well as in patients with no heart failure. And for example, for the heart failure-free patient, you can imagine, like, say, if patient is diagnosed with, diabetes, right? What is the risk of that patient having heart failure late in life? And for patient with heart failure, the setup is typically different. The heart failure diagnosis is served as an index event in this case, and so the question becomes, what is the risk of having, like say, rehospitalization or repeat heart failure, worsening heart failure, or maybe risk of death, or the incidence of some other diagnosis related to the cardiac function. So those kind of the models you can see in what's being published. So for our study design, we considered including research studies that are published in English, available on PubMed from 2019 to 2023. So I'm doing 2018 because Ditana looked at publications prior to 2018. So that's why I'm kind of looking at that particular chunk. And because it's also sort of most recent publication focus, and so it reflects sort of current tendency, right, in the field. We're looking at prognostic studies in heart failure patients. We're also looking at diagnostic studies in patients with and without heart failure, so all kind of flavors of prediction studies. We look at any patient health-related outcome or diagnosis, and the data source is pretty much anything. So it can be randomized clinical trial, registry cohort studies, and any methodology. So we don't really care what kind of methodology was applied, so we just leave it all. We exclude all the studies that are not related to heart failure in any way. Studies that may be related to heart failure, but mostly focused on validating existing models, because that's kind of a different question that should be asked there. Studies that involve biomarker discoveries, again, a very different line of modeling. Studies that develop methodology, like for analysis, more complicated analysis of data. Longitudinal dynamic models, we excluded those as well. Added value of a single factor, so if you have a prediction model, then adding a particular factor does or does it not improve the model, so we can look at that. And then also meta-analysis reviews, briefs, and reports. So that's what had been excluded from the study. And so our approach is pretty simple. Well, what we want to do is read through those papers, abstract the data from those papers. Now, which elements to abstract comes from what's outlined and recommended in Probust and Tripod. So those are two tools for assessing and reporting modeling studies. And I'll tell you a little bit more about that. And what we really wanted to focus is on analysis methods, right? Because there is a lot of ways the model development can go wrong. It can go wrong because you didn't define the outcome correctly. It could go wrong because you sort of massage your response variables too much. It could go wrong because your definitions are inconsistent, right? But to make that kind of an assessment, you really need to be an expert of the clinical domain. And since we're just a bunch of statisticians, we said, well, we cannot really assess that part. We just trust that that part has been assessed by the editors. We just want to see how they developed, how do they go about developing predictive models, and what sort of assumptions, methods, and calculations and reasoning went into that, right? So I'm not going to talk about the electronic health records. Again, that's like a whole big animal in itself. A lot of sort of difficult, complicated, and accurate data there. So we just take it for what it is and just focus on methodology. This is a very nice, helpful chart with predictive study designs. It's republished in Collins et al. So Collins and colleagues published this 3 papers on evaluation of clinical prediction models. It's like fresh off the press, 2023, 3 parts models at BMJ, I think. So DMV in the stands for development of validation, right? And the first sort of approach to developing and validating the model is using, say, the whole data and then validating the model on the same data, or validating it using resampling technique as a cross-validation of Bootstrap. Or if you have clusters, so you sort of develop on one set of clusters and validate it on another set of clusters, right? So that's DV in parentheses. The second one is slightly different way of developing the prediction model. So developing on a random set. So when you have a data, you split it into development data, validation, and then testing data, right? Or if you have, again, some kind of natural subdivision within your data is a geography, so time may be developing on earlier data and validating on later data, for example. The third way of developing and validating is if you have development done in one data set and then use the external data set for the validation of your model. So that's kind of like the best and the brightest option F. And then the option A is really the worst, right? Because you have the same sample, you develop your model, you validate it, so your performance are really optimistic, and chances are it will not take extended scrutiny, especially if your sample size is pretty small to start with. So like I said, for the model elements, for the analysis elements, we relied on the tripod and the probability. So the tripod statement stands for the transparency reporting multivariable prediction models for individual prognosis or diagnosis. These are really guidelines as to how to write the papers. They were put together by a group of researchers that thankfully included statisticians, also included epidemiologists, clinical researchers. So it has multiple sections like title, abstract, introduction, discussion, and then it has this extensive list of important methodological questions that you have to discuss when you write a paper, right? For example, you have to give all the proper definitions for the cohorts and the outcome. You have to justify the sample size being chosen, how you treat the missing data, what analysis you use, how to find risk groups, how you validate, and so on and so forth. And the second tool that we use to kind of identify the elements that we want to abstract is from the prediction model risk of bias assessment tool. So that one consists of 4 main domains: participants, predictors, outcomes, and statistical analysis. Altogether, there's about 20 questions, and the questions are only can have one of the 3 answers. So it's like yes, probably yes, no, probably no, or not enough information, right? So it's a very qualitative tool. It's sort of, does the definition seem to be right? Yes, probably yes. Does the outcome seem to be defined correctly? Yes, probably yes. So it's not like a very exact tool for measuring bias, but it's sort of the best that exist on the market, so to speak. So tripod guidelines is just guidelines for the reporting, right? This is more for assessing the quality of the development. And so here again, we are not looking at the first 3 domains because we are not qualified to assess the definition of predictors and outcomes in participants, but we're looking at domain elements that correspond to statistical analysis. Because as you know, if you use inappropriate methodology or if you do too much data massaging, right, that can all affect how the model will be performing at the end of the day. You guys have any questions so far? Okay. All right, so here the variables that we abstracted from the study. The first one is very basic study information, like who published, when they published. Study design was its diagnostical perspective, population, location of the study, so location of where they were collected. For the models itself, we look at the outcome, we look at the sample size, number of events, and method that was used, how the missing data was handled, and how data complexities were handled. And so question 4.4 and 4.6, as well as like 4.2, 4.5, so those are all the probust elements, right? So they came from the probust tool, and the rest of them is kind of like a mix of tripod, probust, and sensible reasoning. For the development, we want to see if the data was split for train and test. If the data was split, was the outcome distribution accounted for or not? What was the sample size used in development? What was the number of events used in development? How many predictors the development started with? That's P sub C. And then how many predictors were in the final risk model that was derived, right? So that's P sub M. Those numbers tend to be different. How the continuous and categorical variables were treated. It's very common for people to modify the variables, and typically they would decarbonize them, right? They would say, oh, if it's above 50%, it's great. If it's below 50%, it's not. And so we don't want to see that necessarily. which is a variable selection from the univariable analysis was avoided, so we don't want to see much of that either. Accounting for overfitting and optimism, and then how the results were interpreted, if the risk thresholds and calculations provided, and so on. So that's for the development part. And then for the validation test and testing, essentially for the testing, there was majority had tests done on the internal subset of data. So this chart was a large data set and then we reserved some of it, and that's what I call internal testing. And there's a few studies who did external testing, which means they developed a model on one set of data, and then they validated this model on a different sort of data. And that second set of data sometimes can come from a different healthcare system or a different registry. It has like no relation whatsoever with the first development set, right? And that's why I call it external testing. And so for those elements, we again extracted the sample size, the number of events, what methodology was used for the validation, was it the calibration or discrimination was assessed, and for the external testing, whether the population was appropriate, right? Because when you test an external population, you want to make sure that you're testing on a population similar to that the model was developed. Does it make sense? Like 3 was the like reasonable choice. Was it very so how did we go about extracting all that? So we have 5 statisticians on the team. And we got all trained in the sense that we reviewed the variable elements, we read through the provost. And then each of us had to evaluate 5 articles. And then I analyzed the result and we sat down together and looked over major sort of contention points where we disagree. And we decided that we discussed why we disagree and how we should approach that particular question. So that is to ensure the consistency of interpretation between the 5 of us. For the provost, a lot of disagreement was about missing and complexity, because treating missing variables and treating the complexity of the models is you think about missing as complexity, but in provost it's addressed into different questions. So it can be not adequately addressed as far as missing values go, but it can be adequately addressed as far as other complexities like time-to-event analysis, right? Time-to-event data. And then with tripod is there's a lot of confusion came from the fact that in published writing, the words validation and testing are often treated as synonyms. And so what we call validation or what we call testing, a lot of publications refer to as validation. And so you have to really read carefully between the lines, because it's not how they call it, it's how they should be calling it, right? That's what we want to expect. So for the data abstraction, we pulled the query from the PubMed. I screened all the titles for inclusion. Then we reduced it to the papers that we thought that should be reviewed. Those papers were distributed randomly between the 5 of us. So each paper was reviewed by a single reviewer. Data was abstracted as assigned. So if the paper reported multiple outcomes, we only choose the main one. If the primary is main outcome not indicated, we chose the outcome most favorable for the paper. So typically it's the outcome that has the largest number of events associated with it. If there are multiple models reported, so similarly, we chose the models that sort of best model developed by the authors, right? Typically the ones with largest sample size. So we try to be as generous to the authors as possible. So we are not picking the worst that they reported in the paper and all of that. And then once all the data was abstracted, I read all the papers and I adjudicated all the data that was abstracted. So essentially each paper was read twice, right, by one team member and then myself. And then for the analysis, so I'm going to tell you kind of what kind of data we got. I'll talk about the sample size for the model development, so what it is and what it should be. And I talk about these 2 different types of prediction models that we call either true predictive models versus imposters. I'll tell you what that is, and a little bit of sentiment analysis that was done on that by the abstracts. So before we go there, I'll just give you a brief reminder for the sample size estimation when it comes to prediction modeling. So you're all familiar how to estimate sample size, right? If you sort of thinking about hypothesis testing, right? So if you design a clinical trial, there's a very particular design, there's a particular test that you associate with the design, and so you design the study based on that methodology that's going to be used in the analysis. So for the prediction studies, the situation is a little bit more complex. A lot of times what you see in the literature is this rule of thumb that's being recommended, like 10 events per predictor. And it's even a question, so on the probus, there is a question whether the sample size is adequate. And it says it's adequate if it's more than 10 events per predictor, right? Then consider it's adequate. Now, this 10 events per predictor is actually 10 events per variable because the number of variables in the model is not the same as number of predictors, right? Because you can have categorical variables that have more than 2 levels, so you have more than a single variable representing that variable in a model. And so we're talking about not 2 events per variable, but 2 events per predictor. I guess I mixed up a little bit here, but what's treatment correct is EPP. Well, the thing about the 10 events per predictor rule, the rule was established not in the context of prediction modeling. It was in the context of bias and accuracy of regression estimates, right? So it's really about inferential estimates when this rule was determined to be somewhat useful. So it has nothing to do with prediction. And after that, there is some workarounds and some additional research was done, and there's a lot of debate about it. And the numbers it's been suggested anywhere from 15 to 50 events per predictor. So that's not sort of the standard when it comes to prediction modeling. There are 3 other alternative approaches, and those are the ones that we kind of going to be using in our research to determine the optimal event sample size. So the first one is based on precise estimate of outcome risk. So if you can think about the risk rule, the minimum thing you want from your risk rule, say if you talk about binary outcome, is on average in the whole population, you should have, roughly the ballpark estimate of the proportion of events in the population, right? It's kind of the minimum desired performance of the risk rule. And so for the binary outcome, if by your outcome proportion, and you have delta is your margin of error that you want to, the precision with which you want to estimate that outcome proportion, then you can derive a sample size based on the confidence interval for that outcome proportion, right? So formula very familiar, you specify delta, once you specify delta, you can write up sample size as a function of delta. And then phi hat will come from your sort of field knowledge about a particular prevalence or outcome disease in certain populations that you're interested in. So this translates to the time-driven outcome. So with the exponential model, you can make use of the confidence interval for the cumulative incidence function and time t. So again, you can get the outcome rate from the historical data. You can specify the landmark time of interest, and then you can estimate t so that the confidence bounds, each bound is kind of within your delta margin from your estimated average, right? And that will give you the sample size. So that's one approach. Now the second approach is to minimize the shrinkage in a model. And so if S is the shrinkage parameter in your model, right, then the formula here that doesn't have a particularly difficult derivation, it looks a little scary, but P on top, so that's the number of model predictors. So it's a number of parameters considered in the model. And then R squared is the performance of your model, right? And so R squared for continuous outcomes is just a regular R squared. And then it generalizes for the binary, for non-continuous outcomes. The only difference for non-continuous outcomes, it doesn't go between 0 and 1. It tends to be a lot less than 1. And it depends on the proportion rate, right? And so here again, if you specify if you think about designing the study to build a prediction model, right, designing a predictive study, you should have an idea about how many model parameters you want to start with. You can get R squared from the say time models or historical models or what have you, historical data supposedly. And then the shrinkage is again something you set up. So typically it's like 0.9, right? So you want to attenuate your coefficients towards 0, but maybe. And that gives you sperm. So that's the second way to estimate the number of samples required for the study. And then the third one, so this one is about not looking at the relative R squared, but looking at the absolute change in R squared. And so we are not looking at the functional R squared in that sort of prime form. We're looking at standardized version, so it's divided by maximum. So now it goes, the Snaggle curve is R squared is going to be going between 0 and 1. You can interpret it as a percent variation in your data, right? In your model. And what you want is you want the difference in R squared based on your development set, so that's apparent R squared, and based on a new data set altogether. You want the difference to be as small as possible, right? Because you want to develop a really good model that when applied to a new data set would perform really like just as good as what you saw in development data set. So you want the difference to be less than delta. From here, you can express your shrinkage value for the shrinkage parameter. And now that you have a value for the shrinkage parameter, you can use a formula from the previous slide to calculate the desired sample size, right? And so those are the three different approaches to calculate the sample size for the prediction model. None of them really used in clinical practice very often, as you can imagine. Like none of them at all. So, any questions so far? Okay, so here is the study data. When we did the original PubMed query, we got close to 6,500 abstracts. From there, we included all the studies that have like animal models, Mendelian optimization, and then didn't have a very specific keywords in the title. So that brought us down to about 2,500 studies. Okay. Is that how much left? Oh, wow.
No, it's 4:15. You're 15 minutes.
Okay, so we done 2,500 studies. So I read through all these titles and excluded, no, I read through all this abstract and excluded a bunch of other studies for various very good reasons. And so we done to 237 studies that are presented in the literature as prediction modeling studies. Now, those studies are not all prediction models. They're a set of true prediction modeling studies, about 108 of them, and then there is a set of what I call imposter studies. So those are the studies that pretend to be prediction modeling, but they're not prediction modeling. They use a lot of prediction modeling language, but they actually didn't build the models the way the prediction model should be built, right? And so reading through the full 108 true prediction study papers, we excluded 25 extra because there's some other good reasons for doing that. And so we're analyzing the 83 studies. And that's what I'm going to talk next. So 83 studies is, this is what we're getting. Oops. So majority of them were prognostic studies in nature. There's 40% came from Asia. I was kind of surprised that there was such a large fraction of studies from Asia. 21% North America, primarily US, but there is a few Canadian studies. Mortality was a very popular outcome. A lot of studies were not specific about the event time. They just look at the risk of events throughout follow-up, which you kind of debate whether or not it's really useful prediction model, right? Majority of studies relied on ensemble modeling and building a prediction model, so random forest, gradient boost, not so much on logistic regression. And there's a little bit of time to event, neural nets, and regularized data. And about 70% of studies use some kind of data split, not necessarily in 3 subsets, but at least in 2 subsets. So popular approach to split the data. So here's a summary for reporting just very basic statistics. So number of samples and number of events. So the samples and events at start is it's how many you had at the very, very beginning of the study. Because if you look with clinical data, with such messy data, you never end up analyzing or using data that you just got from the analyst. There's always a lot of processing involved. And as you can tell, not all of that is reported. In particular, the number of events at start is rarely reported. And actually, the number at start is overestimated, right? So the parameters at start that are being reported are slightly overestimated because a lot of publications didn't even report that they started somewhere. They just say, oh, we have these 5,000 patients and we're analyzing them. And so the impression is that they've done absolutely nothing to the data, that they got this really nice clean data and just went with it, right? But as you can see, so for this basic 4 parameters giving a lot of benefit of the doubt to the authors, not a single one of them. Well, actually, only like 40% of them report all the 4 parameters, right? So pretty low reporting rate. Now, when it comes to modeling, so if we look at the summaries based on the robust elements, so just to look at this picture visually, what you want to see is a lot of green color, and green color means that it was a good answer, like yes, probably yes, that they did it right. And you want to see a lot of this color, and you don't, right? You see a lot of like red and yellow. There is 7 elements here, and not a single paper hits all the 7 criteria, right? There is only 3 papers that successfully hit the 6 criteria, but not a single one hit 7. So for the most part, people avoid manipulating variables too much or using univariable analysis. The biggest issue to me was how inappropriately missing data was addressed. And then the estimating or reporting relevant performance measures. And I'll tell you a little bit more about that. And so not accounting for optimism is kind of part of that picture. So based on that, it's, it's not a low risk of bias type of studies. So for the model development, again, it's so surprising to me that only 60% of the studies actually report how many events they had in the data when they develop in the development set. They can report the whole set, but not the development set. The number of predictors considered was, reasonably high, so the median 51, and then went down to roughly 13 after the model was developed, so that was expected. How to use a model was not often discussed, so they typically give some model performance parameter, but they don't really tell you what thresholds to use or how to apply the model. And only a third of the papers had some kind of information that would actually let you to produce a risk model. It would be like a model explicitly, or maybe a normal count chart or something like that. So when we looked at the sample size, so we simulated we abstracted the sample size that we used in model development, and we also simulated sample size that should be used if the authors properly designed the prediction model studies and calculated the sample size that needs to be used, right? So the first plot shows you sort of how it should be if we truly consider the number of predictors before the model is built, right? So you at the beginning of the experiment, you think that you're going to have that many variables that can possibly be candidate for your prediction models. And based on the chart, right? So there's a vast amount of studies that actually, 71% of them that had the sample size that was too small than needed for the model development. Now, picture looks a little bit better if you change the number, the P, right? So rather than the number of predictors at the start, you say number of predictors at the final model, right? Because then you have a lot fewer model, a lot fewer parameters, and so you don't need as many samples. And so now you actually kind of maybe not too bad, but still, like, half of them are doing all that good, right? Even in the most favorable sort of a condition. So that's kind of like a big red flag here. Model validation, cross-validation super popular as you may have expected, 70%. Calibration almost never done. And we had the calibration mentioned, means if you just say the word calibration or we did calibration, we count it as you did the calibration, right? I mean, at best, we saw the plots. Not a single one sort of tried or even attempted to do any kind of optimism adjusted in the model. AUC for the developed model almost never reported, like 53% report. Well, I get half of them report, right? Like very, very low reporting rate. So for the model testing, the internal and external. So for the internal testing, so again, this is when you have a whole data set and you reserve some of it for the internal testing. You have a number of events, not always reported, so 70% only. Everybody chooses to put the AUC for the most part, and again, no mention of the calibration. And then for the external one, was really surprising because it was only 15 papers, right, that did the external validation, but all 15 of them reported the calibration. It was kind of like really sort of incredible, because if you think about it, like it's kind of going an extra mile, like why do you need the external, right? So it's really like an extra level of complexity in your study. And so people who go there, they kind of understand what the prediction modeling is really taking, and that's why they are developing that. They are reporting discrimination and the calibration for the models. And so briefly about the sentiment analysis. So now I'm looking at the all the imposter abstracts and all the true prediction modeling abstracts, right? And so imposters typically report inferential models, but they use a lot of predictive modeling language. They say predict predictors, and it will help to predict and so on, right? So you read it, and if you don't know any better, you say, oh, they know how to predict the risk of event limitation. And so what we do is we essentially look for all the comments, so, for all the words, right? So we abstract all the words from all the abstracts. We filter out all the comments of words, do the count matrix. We also limitize the words, so like say models and model, they kind of mean the same stuff, so we treat it as a single name. Remove words that occur not at all or occur in all the papers. And then we do the clustering essentially using the Latin digital allocation model, just to see if we can identify those two groups of papers, the imposters and the true prediction models, just based on how the abstracts are written, right? And then we couldn't help because we're in a prediction modeling. We also do see if we can build some very simple prediction model based on that. And so the interesting part was that the vocabulary that was used in the imposters and the true prediction model was pretty different. Like the most surprising part was the word model was used a lot more in the true prediction modeling papers than it was used in imposter papers. The two papers, right? So what you see on the left is the probability of being in cluster 1 versus cluster 2 versus the expert classification. And it was just incredible for us how well those two corpus of abstracts were separated, right? It seems like based on the vocabulary that was used just in abstracts alone, you can actually make a very clear determination was it a true prediction modeling paper or the imposter. And the random index for that part was 0.76, which is pretty high. Now, random forest, if you just take the count matrix and feed it into the random forest using, I think, the first two principal components of the data matrix, you actually get reasonably good discrimination power. Again, it's probably overestimated, but it tells you that it may be possible to have some kind of tool to help identify those papers. I mean, as an editor, I can tell you it would be very helpful. So that's kind of where that comes from. And the last part is about the hype language use. So that's an official terminology in linguistics. So SPIN is a trend in writing strategies, and that includes peer review studies. And SPIN is a specific intentional or unintentional reporting that fails to faithfully reflect the nature and range of findings and could affect the impression the results produce in readers. Okay. Things like selective reporting, glossing over some findings, ignoring negative findings, claiming results that are not justified by analysis. Those are all part of the SPIN, right? And there is a specific mechanism to implement SPIN. It's hype. So hype is what words you use to spin your work. And so hype is a hyperbolic subjective language that may be used to glamorize, promote, exaggerate your research. And that hype has a very characteristic lexicon. So in 2022, Miller, Battal, and Gudgel, they published a paper on high words vocabulary. So Miller is a linguist and Gudgel is a biomed researcher. So they extracted about 900,000 plus abstracts of NIH grant applications. And they went through the body of abstracts and they identified 139 hype objectives, right? And that's what we're using as hype classifiers. And so if we look at 139 hype words, we have very few that actually showing up. Like we have 17 words that appear once, 8 that appear twice, and 9 that appear 3 times. And then if we look at the distribution, right, this is significant, but sometimes you can say, well, significant is appropriate. It's not really a high word, it's a true prediction model. But then there is like words like accurate, vital, on top, shows for the true models, and then powerful, advanced, strong show for the imposters models. That was kind of like an interesting how those two reflect. And when we look at, so these two radial plots show the, so the black line is our models, like the data that we reviewed. The red lines is the 2020 rates from the paper that originally developed the hype language adjectives, and the gray line is also from that paper that developed the hype language adjectives and 1985 NIH abstract applications, right? And so you can see that what was originally found, it was found that the rate of 2020 hype language is higher than 1985. And what we see in our paper that it's not getting any smaller. So the rate of the heart failure papers that we reviewed is comparable, right, to more to 2020 than it is to 1985. The counts were too small, so we didn't do any formal analysis, just some visualizations. So what do we have kind of at the end? So we have from 83 true prediction model studies, we had a single study that used tripod for guidance. We had 0 studies that adequately addressed all the robust items that we assessed. We have less than half studies that actually report event and sample sizes to begin with. 90% not handling missing data correctly. 70% have sample size below the required minimum. 63% not assessing the calibration in your model. And only a third, and maybe it's a blessing in disguise, right? Maybe the third of them is available for use. From 83 studies, it was like 1 near perfect paper. It was like, I think it was like 1 reliant. No, but like seriously, right? Like it was such a beauty to read. It was like so like I have a separate folder where I say like good paper, so it's in there. And so what it seems from that exercise we just just presented you is when it comes to prediction modeling, the study design, analysis, and reporting is really subpar. There is a lot of inadequate development and bad practices. And that fundamentally can lead to incorrect, potentially harmful clinical decisions. And we have half of the papers that we identified actually not the true prediction model papers, they're imposter papers, right, that are masking as true papers. Most of the developed models are not actionable. So if you ask me if it's like signal or noise, I tend to go like, it's probably noisy, and maybe it's a good thing that it's not well used in practice. So there, I think did I give the time?
Perfect.
All right.
Does anyone have any questions? How can we somehow find the criteria that would be the model? When you say that 90%, for example, they fail at a certain criterion, or 70% will have the proper sample size, how are we able to know what is the proper sample size? So can you just repeat the question?
Oh, okay. So the question was, how do we find the proper, like how do we know what to report for a good study, right? Is that what you're asking? So tripod is actually an excellent checklist. So tripod is a checklist that was put together by statisticians and like good very smart people, and it hits all the important elements. It literally tells you, you have to explain this, you have to explain that. And they just published a new tripod version for AI. So the current tripod version is for regression-based methods, and this one that came out is for AI, so like machine learning and stuff. So I would say if you want to report fairly well, read things like, read the robust, read the stroke. So it's another checklist, but it's more like for epidemiological studies. And then the tripod if you're doing the prediction modeling. Yeah, it's an excellent question. And then find a paper you really like and see if you can see all the elements in it. And then follow the example.
Larissa's perfect paper.
Yeah, almost perfect.
I'll have to ask you later on this. I don't think we're gonna ask that today, not recorded. It looks like there's a question in the chat. If I thought that correctly, is whether or not your work has been published?
No, not yet. It's like we'll literally. Yeah, I just finished. I mean, some of it, but I work in progress. But
Are there any other questions? So I wanted to ask, in terms of how, can you say a few words to how the published paper will translate to actual medical practice? Because that's a bit of a
mystery.
yes.
Okay. Yeah, it's a very good question. It's really, was it translated? Some of them do.
Personally, I've seen like after I've been exposed to a medical practice, I've been able to track down the paper where it's published, sort of the study or the results, but I've never gone the other way, like how often do they actually?
I think the adoption rate in clinical practice is pretty low. I think right now FDA is just coming with guidelines as to how to make AI or machine learning based diagnostic model enter the clinical practice. There is that. I think a lot of times clinicians just want to know this information as a helper, like just to see if that helps them to understand how better treat this patient or monitor this patient and so on. So making this any predictor model official, like integrating it with say patient records, the way you come in and you press the button and it tells you the risk score. This is a huge problem. That it's a huge undertaking. It's very difficult. You need to have the FDA clearance, and if you do, it's like they just want to get not as a diagnostic tool, but a diagnostic aid. So it cannot be the only thing that determines your clinical decision. It has to be something that helps you. That's like the easiest pathway to get there, right? So there's a number of different scores used in practice. I see a lot of the scores used typically in research, right? That they always use some kind of measure of baseline health of a patient. But to say that you can bring it over to the bedside, I think if it's broad, it's sort of in a more unofficial capacity, so to speak. Some of the scores that are being implemented, they available online. So doctors can always go and do one of those online calculators and calculate like tears or minute score or whatever. But I don't know how many of them are officially part of the. I don't think any of them officially. Like in our system, we don't have a single risk score that is. No, there is an STA. Yeah. So it's like maybe a couple of scores that kind of been adopted over time, but they kind of report it as a parameter, and then not necessarily something that just guides decision making. So it's a big kind of complicated question. Later later.
Well, I think I would say that you're very much tackling a complicated issue.
Well,
you're poking the bear.
Yeah. Shining the light.
You're poking the bear. Yes.
Yeah, it would be interesting, but it's hard to get. But it would be interesting to compare these with the rejected ones. What happens with the rejected ones?
The papers that didn't get published.
Yes, why they are not published? Is it the trend similar? You will get similar percentages, or there is some difference?
Yeah, that would be interesting, right? But it's like,
I think.
it's the one you.
But you can check like the
you don't know.
archives and see.
But there's one thing to be said, right? If the paper doesn't, because we didn't kind of tier the papers here, but when the paper is not published in a good journal, it eventually ends up finding a kind of a low tier journal. So that's kind of unfortunate because really nothing changes in the paper. The critics stay, they just find another paper. So in some way, they're in there, like in that mix, right? There are those rejects that then found the second home. So it's a good question, though. I don't know.
Especially with the language. Even language can be a determinant to reject a paper.
You know, I have to say, I was really surprised by the number of studies coming from Asia. And those studies actually tend to be pretty good quality. They tend to be pretty large studies, right? And so they often based on large medical records, sometimes multiple hospitals contributing, and they typically have some computer science element around it. What I think the problem here maybe is there's, peer to publish, everybody's doing it, so I want my own prediction model. I want my own AI on my title talk, and then just kind of not collaborating with statisticians. And big part of it is how easy and easy to use and accessible the statistical software is these days. I can tell you, like in our organization, we have scholars who just take the data, feed it in, and get the results out. And sometimes they come with questions, and then they have to go back all the way to the beginning because there's like this certain element that was overlooked, right? But because the software is so easy to use, people often avoid finding a statistician. My probably 2 out of 3 papers I return back saying, please go consult a statistician. So that's it's a big problem in the field, and I think we have to be kind of active advocates about that. So I would like all encourage you to like look out for some opportunities to be statistical editors and review them, contribute them that way. Because it's the only like our profession is taking over slowly by this data scientist, operative, right? AI and so on. But really, when it comes to experimental design and modeling, I think we have a lot of strengths, right, in our tool set, and that's what we can bring to the table and kind of shine the light on that. So don't be swayed away by buzzwords and like big, big titles, but yeah, I think that's where our mission should be.
Yes, well, I think that is a good summary. Leaving everyone with the mission, so please join me in thanking Larissa for a really interesting and important talk.